All right, so we've got a few people in, so we'll go ahead and just get started here. So uh, this is kind of the way we're going to be doing lab for the kind of for here on out for the rest of the semester. So we'll do kind of a short pre-lab, kind of a pre-lab lecture. I like to say around 6:30 every night, or 6:30 on Tuesday nights. So at our normal lab times, we'll be meeting on Zoom, and I'll send out Zoom invitation so you can have access to the email or to the link, and then we'll kind of go through our lecture, and then after that's done, I'll Zoom will kind of record the video. I'll upload it onto my computer and then upload it onto YouTube, and then take the YouTube link and I'll add it onto Canvas. So you'll always have access to those Canvas pages and the lectures before, I guess not before, but after every lecture. Okay, so if for whatever reason you're not able to log in or watch the pre-lab lecture live, you'll always be able to come back later and listen to it again. Okay, so that's typically the way this is going to go. And then there's going to be a lab assignment for each lab. I'm kind of going to be modifying some of the stuff that's in the lab manual in order to help us do things in a more online manner. We get a little bit complicated sometimes, but I think we'll be able to manage. Cool. Any questions on how things are going to be going so far? Kind of type into the chat if everything's okay, or if you have any questions about anything. Okay, so if not, we'll kind of just go through our what's going to be our normal routine, and then if you have any questions after that, I can explain anything. Okay, so what I'm going to do is I'm going to go ahead and take over the screen, and I'm going to pull up the lab PowerPoint for this week. Okay. All right, so is everyone able to see the PowerPoint?
No.
No, not yet. Okay. All right, hold on here. All right, how about now? Did it come up?
Yeah.
Yeah, okay. Okay, cool. All right, beautiful. So Zoom is going to be a little bit tricky from time to time, but I think we'll be okay. Okay, so for today's lab, we're going to be going over muscle physiology. Okay, and so have you guys talked about muscle physiology already in a lecture? Yeah, okay. All right, so hopefully this should all be kind of sort of review, and then we can kind of go through a little bit of this as well. Okay, so we're going to be looking at muscle physiology today, and so we're going to kind of go through the different components for a normal muscle contraction to take place, and then we're going to look at how we're able to actually measure this in a lab setting. Okay, so to start off the entire muscle contraction, we're going to begin at the neuromuscular junction. Okay, so at the neuromuscular junction, we're going to have a somatic motor neuron, and the somatic motor neuron is going to synapse with a muscle fiber. Okay, and so to trigger, I guess initiate a muscle contraction, the first thing that we need to do is we need an action potential to come down the length of our axon. Okay, once we have that action potential or our nerve impulse reaching our axon, okay, that's going to cause the voltage inside the axon to change from being negative to positive. Okay. And so cells are typically negatively charged, but when they have an action potential, there's going to be an influx of sodium ions, which are going to help increase the voltage inside the cell. And now when that voltage changes inside the neuron, that's going to open up these voltage-gated calcium channels, okay? These channels right here. Okay? And so when our voltage-gated calcium channels open, that's going to cause calcium to flow into the neuron. Okay? And once we enter the neuron, we're going to enter the synaptic end bulb of our neuron. We're going to find a bunch of these synaptic vesicles. Okay, and all these synaptic vesicles are going to contain a specific neurotransmitter that's involved with muscle contraction, and that neurotransmitter is acetylcholine, or ACH. So I'll kind of draw that here real quick. Okay. So these neurotransmitters, this is ACH, acetylcholine. Okay, sorry, this is a little bit ugly, but kind of bear with me. Okay, so that's acetylcholine inside of our synaptic vesicles. Okay. So when calcium enters the neuron, that's going to cause the synaptic vesicles to undergo exocytosis. And they're going to start releasing that acetylcholine into the synaptic cleft here. Okay. So once it enters the synaptic cleft, acetylcholine is going to start binding onto these acetylcholine receptors. Okay. And when they bind to the acetylcholine receptors, the receptors will then start to act as sodium channels. And so they're going to start to open up. Once they open up, they're going to allow sodium to enter the muscle. And when sodium enters the muscle, that's going to trigger a muscle action potential. Okay. So that's going to be the first step. Okay. The neuromuscular junction, we're going to have an action potential. That action potential is going to cause these calcium or voltage-gated calcium channels to open. They're going to rush in, and then calcium is going to trigger exocytosis of the synaptic vesicles, which contain acetylcholine. Okay. Acetylcholine gets released in the synaptic cleft, binds to these receptors, and then the receptors will then open up and then begin acting as sodium channels. Okay. So once they open up, sodium is going to, and once it enters the muscle fiber, we're going to create a muscle action potential. So, this sound familiar? Is there any questions on the here? Are we okay? Okay, so it looks like we're okay. Okay, so the next step at the in order to trigger a muscle action or trigger a contraction is when we produce that action potential in the muscle fiber. Okay, the muscle fiber is going to or the action potential is going to run along the length of that muscle fiber, along the sarcolemma. Okay, and then it's going to start dipping into these T tubules. Okay, and so these action potentials are going to continue running along the entire length of the muscle fiber. Okay, when that happens, okay, again we're going to have a voltage change, and that's going to cause these voltage-gated calcium channels inside the sarcolemma or inside the muscle fiber to start opening. Okay, when they open, they're going to cause calcium release channels to also open, and those calcium release channels are going to be in the sarcoplasmic reticulum, as we see over here. Okay. And so we're going to end up getting extra calcium getting released from the sarcoplasmic reticulum due to these action potentials here. Okay, so once calcium gets released, it's going to start binding onto a specific molecule on the actin filament, which is called troponin. Okay. And so when calcium binds to troponin, it causes troponin's formation to change. It changes its conformation, and that's going to pull tropomyosin away from the myosin binding sites on actin. Okay. And that's going to allow myosin and actin to now create these cross bridges. Okay, so we'll look at that in a little bit more in a kind of a more detailed picture here. Okay. And so these calcium channels are going to stay open as long as these action potentials continue running along the length of that muscle fiber. And also, those are the different components there. Okay. So just as a kind of a bigger picture here, before we release any calcium, okay, we're going to have this molecule or rope called tropomyosin, and it's going to be bound, or it's going to be kind of covering up the myosin binding sites on the actin filaments. Okay, and so this is going to be happening when the muscle is relaxed. Okay, and so sitting on top of tropomyosin, there's going to be this smaller molecule, smaller proteins called troponin. Okay, and so when calcium gets released, what calcium will do is it'll bind to troponin, and when it binds to troponin, oops, sorry, there's still extra diagrams here. Okay, so when calcium binds to troponin, okay, it's going to pull, it's going to cause troponin's shape to change, and when its shape changes, it pulls tropomyosin away from the myosin binding sites on your actin filaments. Okay, and so now your actin is now available to bind onto myosin, okay, and then be able to trigger the contraction cycle. All right, the last step that's going to happen here is going to be the actual contraction cycle. And so this is going to be where myosin is actually able to bind to actin. Okay. So what's going to happen first is we're going to have an ATP molecule that's going to bind onto myosin. Okay. And then myosin is going to break down that ATP into ADP and a phosphate. Okay. When we break down ADP and a phosphate, okay, that changes the conformation of the myosin's head, and it shifts to the cocked position. Okay. Once it shifts, it's going to be able to form a cross bridge with actin, okay, to form the cross bridge here. Okay. And then after we form the cross bridge, we're going to release phosphate. Once that phosphate gets released, that's going to trigger the myosin head to undergo the power stroke, and that's going to pull actin in the direction of the M line, okay, towards the M line of the filament here. Okay, so we trigger the power stroke. Okay, now myosin is going to eventually lose its ADP molecule, but it's going to stay attached to actin until a new ATP molecule binds. Okay, when that new ATP molecule binds, it's going to detach from the actin filament, and then it's going to start this whole cycle all over again. And so this is where we actually have our muscle contractions actually taking place. Okay. Any questions on any of these three processes that are involved with a muscle contraction? Okay. So these are ones that you will for sure need to know for lecture, and then for essentially for lecture. This is going to be really important process going forward. Cool. Any questions at all? So after this question is, how long does this process take? This entire process from the neuromuscular junction to when myosin actually moves happens in a fraction of a second. Okay? So this took me a long time to explain, but when it's actually happening, it happens extremely quickly, like within like milliseconds, probably even less than milliseconds, I would probably imagine too. So it's incredibly quick. Yeah, very good question, Ashley. Any other questions here? Okay, cool. All right, so that's how we end up forming a muscle contraction. Okay. Now, in order to create a muscle contraction, we need a somatic motor neuron. Okay. And that somatic motor neuron is going to innervate multiple muscle fibers or muscle cells here. Okay, so here we have one motor neuron, here we have a muscle fiber, here's another muscle fiber, here's another one. And so as we can see, this one motor neuron is going to be able to innervate and stimulate multiple muscle fibers kind of in this bundle here. Okay. So when we look at a somatic motor neuron, okay, and all the muscle fibers it innervates, we call that a motor unit. Okay. And so a motor unit can include anywhere from 10 to 2,000 muscle fibers, okay, as a whole. So, and it kind of depends on the types of contractions that muscle is going to be able to do. So when we're thinking about any kind of muscle contraction that we're able to perform, or any kind of motor movement that we're going to be doing, okay, when we're thinking about any kind of motor movement, this is going to involve the activation of multiple motor units. Okay? So even just kind of snapping your finger or raising your hand or typing on a computer, these are going to involve multiple motor units. Okay, multiple motor neurons as well as multiple muscle fibers, okay, are all going to be involved to help create any kind of motor neuron or motor movement. So this can be anywhere from hundreds to thousands of different muscle fibers all being activated just for a normal muscle contraction. Okay. So depending on the type of muscle contraction that you're going to be doing, you're going to be recruiting fewer or more muscle fibers inside your motor unit. Okay. So for example, if we're doing something like more fine motor movements, right? So if we're kind of just like snapping your finger or writing up on a piece of paper, those are going to be more fine motor movements. Those are really detailed motor movements. Okay? And so as a result, those motor units that are going to be involved with producing those contractions have to have just a few muscle fibers. Okay? The fewer muscle fibers you have, the more fine control you're able to exert over what kind of the motor contraction you're able to do. Okay? Now, when you want to do more gross motor movements, more large-scale motor movements like raising your hand or kicking a ball, okay, that's going to involve motor units with much higher numbers of muscle fibers present. Okay? So the more detailed movements you're trying to perform, the fewer muscle fibers in your motor units. Okay? When you have a larger muscle movement or a larger motor movement, you're going to have more muscle fibers inside your motor unit. Okay? Does that make sense? All right, cool. Okay, cool. So no questions. So now that we've kind of looked at how muscle contractions are going to be taking place, we can kind of figure out how we can measure our muscle contractions. Okay? So the first way that we can actually see muscle contractions in action is if we're able to stimulate these muscles using electric shocks. Okay? And so what will happen is if you take a muscle and you add an electrode to it and you trigger one electric shock, that is going to cause that muscle to contract. Okay? And so what will happen is if you just add one shock, one electric impulse to that muscle, you're going to end up with a muscle twitch, okay, where you have just one single little contraction happening in that muscle. Okay. So that's called a muscle twitch. And so you'll see on a chart here, we're going to have a little bit of tension. Okay. We're going to have some tension increase, and then once it starts to relax, okay, the tension is going to decrease. Okay. So that's going to be a muscle twitch. Okay. The next type of or series of contractions that we can have is called a summation. Okay. And so a summation is where you stimulate a muscle several times without giving it a chance to fully relax. Okay, so for example, here we have a muscle contraction or an electric shock where we have the muscle begin to contract. It relaxes a little bit, and then you hit it with another electric shock. And so the tension begins to add. Okay, you end up building on that one contraction with another contraction, and the tension becomes a little bit greater. Okay, then again, you give it a little bit, and then you hit it with another contraction before it has a chance to fully relax. Okay, and so you start to build on that. Contraction, and you end up with a stronger tension inside that muscle. Okay, so that's what we call summation. Okay, because all of the contractions are kind of summing onto each other. Okay, you're adding extra tension onto each individual contraction. Okay, the last type of muscle contraction that we can have is called tetanus, and so tetanus is where you stimulate a muscle over and over and over again with very short periods of relaxation. Okay. Or no periods of relaxation. Okay, so the first type is what we call an unfused tetanus. Okay, and so here we hit it with an electric shock, we get a contraction, okay, and then we hit it with another electric shock. It relaxes a little bit, then you hit it with another one, then you hit it with another one, and another one. Okay, so it's relaxing a little bit in between each individual shock. Okay, and so this is what we call an incomplete tetanus or unfused tetanus here. Okay. The next type of contraction that we can have is called fused tetanus or a complete tetanus. And so this is where you just continually shock the muscle over and over and over and over again without giving it a chance to relax at all. Okay? And so you have a contraction, okay, and you continue to stimulate the muscle with that contraction. And so as a result, the muscle continues to contract and contract, contract, contract until you stop. And then you get it to relax. Okay, so we have twitches, we have summations, and then we have tetanus, where we can have unfused tetanus or fused tetanus. Okay, and so this is what we're going to be able to see if we're looking at it, or if we're trying to electrically stimulate a muscle and looking at a chart on the tension here. Okay. All right. Lastly, here we're going to be looking at electromyograms, and so electromyograms or EMGs are these tools that are able to allow us to record electrical activity that's happening in a specific motor unit. Okay, and so what will happen is this EMG will have an electrode that's inserted either into or onto a muscle. Okay, and then what's going to happen is we're going to start stimulating that muscle using an electrode. Okay, and so what's going to happen is the somatic motor neurons are going to start to fire. They're going to start to create action potentials. And so the job of the EMG is to pick up on those action potentials. Okay, and it's going to cause them to appear as these spikes on the EMG trace. Okay, and so each of these action potentials and each individual action potential will come up as a specific jump or a specific kind of trace on your trace here. Okay, and so when you have a stronger contraction, you'll typically have higher spikes inside your EMG trace. So when we're looking at contraction strength along an EMG, there's going to be two ways of measuring strength. Okay? The first way that we can measure strength is by looking at how frequent the motor unit is firing. Okay? So if you're looking at an individual motor unit, the frequency that it fires will tell you that you're going to be creating a stronger action potential, okay, or stronger contraction. Okay? So the more action potentials are happening inside that motor unit, okay, the more frequent that muscle is going to be contracting, and so you're going to end up with more frequent EMG spikes like we see over here. Okay, so that's going to be one way of determining strength, okay, by looking at the frequency of the motor unit firing action potentials. Okay. The second way that we can measure contraction strength is by looking at the number of motor units that are firing at the same time. Okay. So if we're using more motor units at once, okay, we're going to end up creating a summation effect because we're having one motor unit being activated, we're having a second motor unit, and all the muscle fibers in it within it being activated, another one with all the muscle fibers within it being activated, and so you end up with a stronger contraction. Okay, so as a result, you end up with a summation effect, and you end up with higher EMG spikes. Okay, so on one case, you're ending up with more frequent EMG spikes if you're looking at an individual action potential or an individual motor unit firing multiple action potentials. Or you can get higher EMG strikes when you have more motor units all firing at the same time, and you get a stronger contraction there. Okay. Any questions on the EMG so far? This is all kind of making sense here. Are there any questions that I can answer right now? Okay. Alrighty. So with that, the last thing that we're going to be able to determine using an EMG is the nerve conduction velocity. Okay. So the nerve conduction velocity is the speed that an action potential is going to be traveling along a nerve. Okay. And so typically, your nerve conduction velocity should be about 60 meters per second, or about 134 miles per hour, because that should be the speed of an action potential going from one node of Ranvier to the next node. Okay. Now, that's what it should be, and so that's going to be determining how, or what an EMG is going to be able to do is it's going to be able to determine the nerve conduction velocity. It's going to determine when the action potential gets triggered, okay, and then compare that to when the actual muscle contracts, and use the two of those to determine the nerve conduction velocity. Okay. So again, your nerve conduction velocity should be about 60 meters per second. However, there are certain conditions that can reduce the NCV. Okay. So diabetes, for one, is a condition that can reduce the nerve conduction velocity just because the nerves start to get damaged there, especially if it's gone untreated. Vitamin B12 deficiency can be a situation that can cause a reduced NCV as well. And then carpal tunnel. And so carpal tunnel can cause a decreased NCV because the nerves themselves are starting to compress. And so because the nerves are compressing, the action potential has a harder time actually moving through the nerve. Okay. Now, so these are different conditions that can lead to a reduced nerve conduction velocity. Okay. Okay. So that's pretty much it for lecture today. What we would be doing if we were in lab is we'd be actually performing an EMG on each other. We'd be able to actually determine our own EMG traces. Okay. I'm going to kind of switch the screen here. And so we would be working with EMGs, and we'd also be working with frog muscles to see when they're going to be contracting at a point in time as well. That's what we would be doing. Unfortunately, because we're not going to be in lab pretty much for the rest of the semester, we're not going to be able to do that. So instead, what I'm going to have you guys do, and I'm going to kind of switch the screen here, is I'm going to show you the lab act for this week. Okay, and so this is it. Is everyone able to see the assignment for this week? Anybody? Yeah. Okay. Cool. Okay, so people are able to see. All right, so that's good. So what I'm going to have you guys do is I'm going to have you guys take a look at 4 videos. The first one involves like what's happening within a muscle twitch. The second one has to do with how an EMG works and what an EMG would kind of look like. And then the second 2 have to do with looking at how adding different substances, in this case adding ATP and adding salt, how that would impact muscle contractions. Okay. And so in these 2 videos, you're going to be looking at some frog legs and how they're going to react when they've been exposed to ATP, a solution of ATP, and when they've been exposed to salt, and what happens to the muscles. Okay. So after you watch all 4 of these videos, so these websites are not posted on iLearn, but the assignment is posted. So this document, I'll show you where that is in a second. So after you watch all 4 of these videos, you're going to answer the questions from the discussions on page, what is it? Page 94 of the lab manual. Okay. So on page 94 of the lab manual, we're going to answer questions 1, 2, and 4. Okay. And that's all you need to do. So this will be due next week in lab. Okay. So I'm going to show you where to go to find this assignment. And again, I'm kind of change the website here. Okay. So is everyone able to see the Canvas page now? Yeah, okay. So here's the PowerPoint. So this is the Lab 11 PowerPoint. And then underneath it, it has the Lab 11 muscle physiology assignment. That's what I just showed you, that Word document that I just showed you with all the websites. Okay? So this is where you go to find out what you actually do. And then to answer the questions, you can either answer that on the Word document directly. You can create a separate Word document, whatever you want to do. Just make sure you complete it. Okay? And then the place that you're going to go to submit it is, so I'm going to kind of take over student view here. So I'll show you how to submit it in a second here. Okay, give me 1 second here. Okay, so to submit it, you're gonna go over to the assignments tab on the left. Once you go to assignments, any second now. Sorry, my internet's a little slow. Cool. Okay. So at the top of this assignments page, you should see any second now. At the top, you'll see a tab that says upcoming assignments. Dang, it is really slow today. Hold on here. Okay. Oh, just kidding. Okay, so we see the lab reports. So you will go over to the muscle lab. Oh, okay, here. Okay, so it does come up with upcoming assignments. So you go to upcoming assignments, you go to muscle lab. And then once you get to the muscle lab tab or the muscle lab page, it will have a button here at the top right corner that'll say submit assignment. Okay. So you'll hit submit assignment. And then at the bottom, it'll ask you to choose a file. So once you save your document under your Word document or PDF, whatever you want to do, just hit choose file, select the file, submit the assignment, and then you will be good to go. Okay. So this will be due next week in lab, next week in lab by 6:30. Okay. So make sure that gets completed by then. And that is pretty much it.